Hey, hello everybody. Good day to you. So, in today's talk, I shall be talking about health-based exposure limits in cleaning validation and basically giving an overview and introduction concerning this particular topic, which has really become topical in the drug manufacturing sector. My name is Dexter Tagire. I'm a professor of toxicology and pharmacy, and I'm a European registered toxicologist and chief consulting officer of HBELs International Consultancy. So, with regards to the learning objectives, by the end of this talk, our participants should be able to state sources of cross contamination during the manufacture of drug products. But I suspect this is something that most of the participants will already know. So, take this as revision in that case. Participants should also be able to describe three historically used methods for setting limits in cleaning validation, as well as the problems with those particular methods, as we shall see in a few minutes. Participants should also be able to state fully the commonly used abbreviations in health-based exposure limit setting. And participants must be able to recognize the methods used in the calculation of health-based exposure limits according to the European Medicines Agency, the World Health Organization, ISPE, SMT. And finally, participants should be able to discuss some of the more important issues and implications relating to permitted daily exposures or health-based exposure limits in their derivation. So this is the more practical issues which we shall talk about in the last part of this presentation. All right. So my presentation shall split into three parts. The first part shall provide a context, which is what I shall be talking about in a minute or so. The second part of my talk will then look at introducing health-based exposure limits from a toxicologist perspective. And then the final part will look at more practical issues, related to health-based exposure limits and how to get these into your own different organizations. So as we all know, cross-contamination is something that is really difficult to avoid. And there is the potential of cross-contamination of a drug product with other drug products which are handled in shared facilities. So whenever I've got a shared facility where two or more products are manufactured, there's always that potential that contamination is actually going to occur. And most of the times this occurs during the manufacturing process, which, as you all know, is quite a long process, which has got several steps to it. So having said that, some more revision. There are several sources of potential contamination during the manufacturing process. And this could come as a result of an uncontrolled release, either from APIs or from other starting materials, of whether it's dust, or genetic materials, gases, vapors, you name it. They can then find their way into another drug product. But contamination could also occur if you've got two processes running concurrently. Two different drug products are being manufactured at the same time. And it is possible that, through perhaps human movement or some other physical issues, a contaminant, an API from one drug product can actually move onto the other one. And another source of contamination is, contaminants from operators' clothing. They move from one batch to another room, etc., and then can actually introduce some contaminants. But one of the major sources that we have, especially in the case of shared facilities, the issue of residues on equipment. And this is where the issue of cleaning validation, which you know about, we shall briefly talk about it, then comes in, where after one product has been manufactured, there's always, some residues. There are always some residues which are left on the equipment which was used to manufacture that first product, and it might actually find its way onto another product. So having said that, the question comes, okay, so what's the problem with contamination? This really should be something which is intuitive and obvious. But, for the sake of revision, we shall talk about it anyhow. So what normally happens is that, when a patient takes a drug and the drug comes with adverse effects, if a person is ill, um, the risk of the adverse effects is always acceptable to the patient. There's always this risk-benefit analysis which occurs. So if I'm ill, suffering from a headache, and I have to take paracetamol, and paracetamol perhaps comes with some GIT discomfort, for me it's an acceptable risk which I can take in order for me to at least have the headache go away. But on the other hand, it is not fair to have a patient who does not want to have that drug in his system because he's not suffering from a headache, in the case of aspirin, to then have the aspirin come into his circulation, causing adverse effects that they would not necessarily have had at the start. So therefore, the risk becomes unacceptable to a healthy patient. And that's a really big problem. And that's the reason why we don't really want any contamination to occur. Secondly, pharmacological effects which may be meant for good in one patient may be detrimental to another patient. For example, if we have a drug, perhaps, which has got vasoconstrictory effects, let's talk about something like ephedrine, perhaps. And then this drug somehow finds its way into an antihypertensive medication where we're expecting to have vasodilation. But because this drug has come in as a contaminant onto the antihypertensive drug, the effects which would have been good for somebody who's suffering from nasal congestion and wanted ephedrine as a decongestant is now moving on to this patient who has got hypertension as a problem, but now the effects are not desirable in this particular patient who's got hypertensive issues. I hope that makes a bit of sense. So just because a drug is good for one person doesn't necessarily mean that it is good for another patient. There could be a drug, bring me to the third item, which is acceptable for females to take if they're not pregnant. But then that same drug could actually be contraindicated in patients who are pregnant, as well as other conditions that, somebody with a peptic ulcer might need to stay away from non-steroidal anti-inflammatory drugs. And now, if the product which they are taking, perhaps for their peptic ulcer disease, maybe they are taking magnesium trisilicate, has been contaminated with aspirin, let's stick to aspirin, that could then have a detrimental effect to this person who's already suffering from peptic ulcer disease. I hope that makes a bit of sense. And then also, there could be some drug-to-drug interactions that can occur with co-administered drugs. So the contaminant could actually lead to drug interactions in a patient who is taking other drugs. And those could either reduce the therapeutic effect of the drug that the patient is actually taking, or could even cause some adverse toxicological type of effects. And that's the reason why, as much as possible, we want to have a drug product, which is free of contaminants, especially if those contaminants are API, which have got a pharmacological activity and also have got some adverse effects, which could then affect this particular patient. I hope that was clear enough. So because of that, then cleaning becomes extremely important. Like we saw, one of the sources of contamination is residues on equipment. The definition of cleaning validation is right there at the bottom of my slide, and it says cleaning validation is a procedure of establishing evidence that cleaning processes for manufacturing equipment prevent product contamination. So it's extremely important, this issue of cleaning and making sure that the equipment that we use does not introduce unnecessary contaminants into another drug product. So cleaning then becomes, going to the first bullet point there, an important risk-reducing measure. You reduce, the amount of drug, if possible, to eliminate it totally, which is really quite impossible, strictly speaking. And, cleaning validation then becomes important. And cleaning validation entails calculation and setting of cleaning limits so as to avoid ill health in non-intended populations. So basically, we're trying to avoid what we were talking about in the previous slide, of the issue of getting a drug which is unwanted into a patient who does not actually need that drug. And that's what this slide is basically trying to let us know. So, having said that, and remembering that today I'll be talking about health-based exposure limits, cleaning validation has been going on for many years, hasn't it? And there have been some methods which have been used before in terms of calculating cleaning limits. And in the next couple of slides, I just want to talk briefly about those methods that we used to use in the calculation of cleaning. Yeah, then after having looked at how we calculated these cleaning limits before, we just want to discuss, for each and every one of them, what was wrong with the way that we were doing things. Why is this HBEL coming in? Why did the European Medicines Agency, put in their guidance? Why did HBEL publish their risk map in 2010? And some of the issues I want to look at concerning the older methods that we used before. Okay. So, two gentlemen published a paper in 1993, and this was Gary Foreman and Michael Mullen. And this basically was then what became the basis of the way that we've been doing our, coming up with our cleaning limits in the pharmaceutical industry. And they basically came up with these methods largely from a food industry perspective, where in industry they were concerned about the issue of pesticide residues in food. The first approach, which they talked about, not really first in sequential order, but one of the approaches that they talked about. In fact, before I talk about this one, the overarching approach which they recommended was that on visual inspection, contaminants should not be seen on the equipment. But then these are approaches which were then doing the calculation. So the first one, based on, it's called the cleaning based limit, is the 10 parts per million. And basically, according to this particular approach, not more than 10 parts per million of a product A, or a drug product A, an API, should be detected in another drug product B. So I'll just repeat that. So basically from that approach, if we had a drug product B, which was manufactured after an initial drug had been manufactured, which is drug A, when we then analyze drug product B, there must be not more than 10 parts per million of drug A in drug B. And basically this was just an arbitrary figure, not health-based, because you're just taking, like 0.01% of the previous drug in another drug, and you're saying that's what you're setting as your limit. And it did not, take into account any pharmacological or toxicological effects of either of the drugs which were being used. Now, the major problem with this, apart from it not taking into account the toxicological properties of the different products being used, was that it was arbitrary, yes. But then, more importantly, it then didn't take into account the issue of potency. And then this becomes extremely important if, let's say, your drug A or your product A is an extremely potent drug, and the one that you manufacture after A, your product B, has got a very low potency. So what that then would mean is that, for your product B, you need, like huge quantities in order for you to get a pharmacological effect. But then the larger the quantity of B, the more the quantity of A. And in the case where A is a highly potent drug, then you can quickly get into the toxicological range of drug A. And as a result of that, this was not really a very good approach to use because of the reasons which I've talked about. Another one which they talked about was the fraction of the therapeutic dose, 1,000th of therapeutic dose. This one was probably a bit better, maybe speaking as a toxicologist, because it took into account the therapeutic, the therapeutic dose, the pharmacology, so to speak, of the drug in question or the potential contaminant. And put simply, what this approach says is that not more than 0.1% or 1/1000th of the normal, ordinarily it's the minimum therapeutic dose of a product, should appear in the maximum daily dose of another drug product. So basically, you got the therapeutic dose, you divided it by 1,000. And what you were saying was that the maximum daily dose of the other drug, which is drug B, where drug A would be the contaminant into drug B, should not be more than 1,000th of the normal therapeutic dose of drug A. And it was quite good in the sense that it did provide, some protection. I put this in quotes. But then in some cases, because you're using this across the board, it could be quite overly restrictive, because it didn't matter whether you were talking about a very potent drug or you were talking about a drug which was not potent. It didn't matter whether you're talking about a highly toxic drug or one which was not as toxic. And as a result, it was being described as being over-restrictive, leading to unnecessary cleaning failures or calls for new analytical method development. And this can be really quite costly for the pharmaceutical industry, where you're developing methods which are supposed to detect very minute quantities for a drug which is probably, from a health-based risk assessment perspective, is not of a toxicological concern. And in addition, this approach, does not consider special groups, e.g. pregnant women, because it's just looking at the therapeutic dose, and not necessarily considering what happens, when that 1,000th, if a pregnant woman is exposed to it as a contaminant in another drug product. And just generally, like I said earlier, a fraction of therapeutic dose was just an arbitrary limit, and there are cost implications, some of which result, from the issue of analytical method development. So it was not necessarily, the best approach, although at least this one did include some pharmacology into it. And we can say in quotes, it did also consider, the toxicological implications, because if you're talking about 1,000th for most drug products, it means from a pharmacological perspective, you'd probably not be expecting any activity to be occurring at those low quantities, and chances are high that for most drugs as well, the toxicological or the adverse effects might be reduced. And interestingly, an interesting study was done, whereby I think more than about 85% or so of different APIs, they were compared, a limit based on this therapeutic fraction of therapeutic dose, 1,000th, compared to a health-based exposure limit. And in most of them, it was found that this was quite well, that the values were overly restrictive when you use this particular approach. But then we'll talk more about this, I think, when I come into health-based exposure limits. The third approach, which was not mentioned by Foreman and Moulin, but has been used in the industry, is to use what is called the LD50. And basically, LD50 is the dose which is required, or the dose which causes death in 50% of animals in a certain population. So basically, the LD50 of the drugs would be used, and then the LD50s would then be divided by a safety factor or an adjustment factor, which could go as high as 50,000. And again, these had problems because it uses death as an endpoint. But there's some adverse effects which have got nothing to do with death. A person could develop a rash, as a side effect, which is far from death. And, it uses data from acute studies, yet there are some other adverse effects which we know develop over time. And from a toxicological perspective, the use of acute data in risk assessment is almost a definite no-no. And concerning this method, there was no guidance in terms of which adjustment factors to use. So not a very good method to use in terms of coming up with the limits. And then here I've just put a table which I took from a paper from Faria, published in Regulatory Toxicology and Pharmacology. Beautiful paper which actually gives some of these issues which I'm talking about with regards to the weaknesses of the methods which were being used in the past. So, just to give an overview of the slides which I've just talked about, industry and regulators have basically moved away from those historical methods, largely because they were seen as arbitrary as well as being unreliable approaches. And they have decided now to move to more scientific approaches of setting cleaning validation limits. And this is a statement that I got off a paper which was published by Shendley, and the website is over there for you to see. And in addition, to the issue of the historical methods being arbitrary and unreliable, there's also the issue of, and they're not necessarily health-based, like the 10 parts per million was not health-based at all, but just arbitrary. And sometimes they could be overly costly, like even the 1,000th of the therapeutic dose could be quite costly if you're dealing with substances which may not be as potent, because now you put the limit so low that you're doing unnecessary chemical tests using advanced methods to try and get to the limit, yet that could have been avoided. Therefore, as a result of that, the European Medicines Agency in 2014 put out a guidance. And there's a statement which they put out there, which I've just copied. They talked about a more scientific case-by-case approach is warranted for risk identification and to support risk reduction measures for all classes of pharmaceutical substances. And this basically is where the health-based exposure limits come into play. Yeah, so I think for now, what I'll just do is to end this presentation here, and then maybe just give some time for discussion amongst the participants concerning the things that we've talked about, any areas of potential clarification, and then we'll pick it up from there when moving on to part 2 of this talk, which will introduce health-based exposure limits. Otherwise, can you just go into different groups and just start talking, about what we've talked about, have discussions. If you've got some questions, some clarifications, write them down and bring them on.